Good evening one and all. In our today's botany lecture, the topic what we have selected, for this I will give some introduction, means some facts, then I will introduce my topic to you today, right? So let's take that facts first of all. I am taking this type of sequence of the topic. Maybe some newcomers, they are not aware about that topic, that is the biotechnology, but properly under biotechnology which subtopic and even what is biotechnology. To reach up to that, I must introduce to you some of the facts. So for that thing, let me to talk about many years ago. I am talking about the 17th century, right? From that time, means can I say many years back, whatever knowledge the human is having regarding what thing? Regarding natural sciences. He kept his full knowledge about this natural sciences for the welfare of the humans, right? Means from that century we are applying all the sciences in the human welfare, means to get best food to eat, to treat our health, to be like healthy throughout our life. All the sciences are working in our comfort and the easy type of the way we are living. But gradually, can I say, when the development happens, gradually many branches of natural sciences introduced. One of that is the biotechnology. In biotechnology, can you see this term biotechnology? Bio stands for the living. Technology means we are applying some technique to get some food or some useful products for ourselves, or you can say for the welfare of the human beings. For example, if I keep here, that is formation of the curd, which is the simplest example I can put. Curd, you know that we are making by using living organism, LAB. What is LAB? That is lactic acid bacteria, right? So we are using the living organism for the production of some useful product. Likewise, we are producing the substances, or you can say products like cheese, even the bread or the dough we are preparing, or any antibiotics we are making, right? So all are the products, or the technique comes under the biotechnology. Now once again, when we are talking about advancements in the science, biotechnology has taken good spin-off. Spin-off means much development happened when it enters to its modern techniques. When the biotechnology became much advanced, then what advancement have been done during that process? Can I say the products, no doubt we are getting from there, but that products to increase its quality and quantity, we have introduced some technique now, and that technique you call it as modern biotechnology. And to get that best of the living organism in the form of organism itself, or in the form of enzymes. Enzymes, why? To make some products from there or running some processes, but all for the welfare of humans. For this, in a modern biotechnology, we are applying the concept of GMO. What is GMO? That is genetically modified organisms. Genetically modified organisms, that you call it as GMO. That will include like plants, or it can include the animals or the microbes, right? Now how this GMO is going to be formed, that is actually the process which you call it one of the principle under modern biotechnology, which you call it as genetic engineering. What you call it as? Genetic engineering. Now this is one of the principle or applications you can say which is making the biotechnology more stronger. One of the part in the biotechnology on which the biotechnology is framed. Now what is genetic engineering? Like you know that engineers, what they are doing, they are assembling a new machinery by taking good parts from the different machines and introducing or inventing such better than that previous type of the machinery. They are developing the advanced one or better one than the previous. Same like that we are doing with the living organism. For example, we are having an organism like A and we are using its cell of organism A. Obviously it is having its DNA. That DNA suppose is having some specific gene over there. Some specific gene is there which is making this as a very good organism. For example, maybe this is a gene which making the bacteria resistive against antibiotics, or this is a cell in a plant where it is giving the better productivity. So instead of taking full plant, now our focus is on that particular gene which is giving us the better result than the previous, right? So this gene we have to take it out. How we will take it out? We will extract out this DNA. We will extract out this DNA from this A cell, and this you call it as a source. Now can I say source can be any microbe? It can be bacteria as well. Microbes include bacteria as well, means prokaryotes, or it can be the plants, or it can be the animals. Now after taking out its DNA, that special gene you have to remove from it, and you will try to put in an organism, suppose B, which is earlier not having that good quality of character due to lacking of such gene. It is having its own DNA, but it is not having that good gene into that. So our motive is to remove this gene from this DNA and putting into this cell. Now this cell you call it as which cell? Host cell, which will welcome this new gene into that and start producing that beneficial thing which is earlier formed by this cell which you call it as source cell, right? But this thing is not happening directly. Like we are having a person suffering from malaria. That patient is transmitting the problem to healthy one through some vector. In the same way here, I am not talking here regarding disease, I am talking about some good character gene which will be given to whom? It will be given to some vector. We are having vector here which is linking the source A with the source B, right? Now this vector is what? This is not an insect. It is what? Usually it is the plasmid. Usually it is a plasmid. I hope so you know what is a plasmid. Like prokaryotes, bacteria is there. It is having its own DNA, but along with its own DNA, they are having extra chromosomal DNA. If this is a chromosomal DNA, this you call it as extra chromosomal DNA, right? Now we are taking this plasmid out, putting this essential gene over there, right? And making such type of intermediate which is having our desirable gene of interest. Now through this vector, this gene is going to be introduced into the host cell, B cell. Now this can be even, it can be microbe according to our product, whatever we need to form. It can be the plant to get the better result in its production. It can be the animals like human beings. If suppose a person is suffering from diabetes, then insulin secreted gene can be introduced into the cell Escherichia coli. Now Escherichia coli will be becoming factories for that. So in that case the host cell will be what thing? Escherichia coli, but it will be benefiting at last what? Human beings. Like animals, I am saying it will be for some another examples. We are now not in the application, just you should know as a rough idea that what this biotechnology is. To take the good genes from the source and putting into the host cell. But linking them between is what? Plasmid, which is a commonly used vector. Now in biotechnology means genetic engineering. What we are doing? We are cutting that. First of all, we are removing this DNA, cutting out this desirable gene of interest, putting into the vector. Now this vector you call it as recombinant DNA. What you call it as? Recombinant DNA. Now you are putting that into the host cell. This all technique you call it as genetic engineering, where you have removed something out from one cell and assembled it with something and putting into something else. Earlier what is happening, if you need something good in a plant, like one plant is having good flowering, other is having good root system but not the good flowering, you have to hybrid it, and then you are getting the two characters in one, and it not so easy. You have to go with the many steps. Maybe the process fails and you have to use the full plant for that technique. But right now we are not with the full plant with the extra what genes we don't want. Now we are dealing only with that specific gene which is giving good quality to that plant, taking it out from that and introducing into the another plant which is not having such type of genes, but through what thing? Vectors. Sometime it will be directly as well, but right now you should consider usually it is done by the vectors. Fine. So what tools we need? Like the engineers, they need some tool to do that process of assembling of new machinery. In the same way here we need the tools. Like tools are required to remove this DNA out. Tools are required to cut this segment of the gene, desirable gene of interest, right? I repeat, what gene I call it as? I call it as desirable gene. You will find in your NCERT book, they are writing even what gene? Foreign gene, right? That gene, why you call it as foreign? Because this is going to be a new gene for this source cell, right? Gene of interest means that gene will produce something new in the host cell and ultimately we get the better products. So we need some tools means enzymes. We need to remove it, we need to cut this, but keep in your mind when we are cutting this out, when we are cutting this thing out, we need the same scissors to cut this plasmid and make a space for this desirable gene of interest, right, and putting that into the host cell. Now what is our today's lecture? Our today's lecture is based on that scissors which will cut the full DNA into small fragment and taking this part separate with along with other fragments. That scissors you call it as what thing? Restriction enzymes, right? So this is our today's topic. We have to talk about the restriction enzymes, right? If I will directly say that biotechnology we are introducing restriction enzyme, maybe the newcomer, those who are crossing through this chapter first time, they don't know what is restriction enzyme. So these are restriction enzyme which are working like a scissors to cut the DNA, right? No doubt we need a specific part, but it will read the full DNA wherever suitable sites are there. What are that? I will discuss within the lecture. They will keep on cutting the DNA at that very points. Then by the other you can say technology, we are separating out the fragments and choosing our desirable gene of interest, or you can say the alien DNA or the foreign DNA fragment we are going to take it out, right. So means this seizures right now, I am talking about the enzyme. Which enzyme I am talking here right now? That is a restriction enzyme. Keep in your mind this restriction enzyme which we are using for our this cell, which you call it as which cell? That is a source cell, and its DNA you have to take it out. What scissors you are using here? Same scissors you have to use to cut the vector DNA, only then this will accept the alien gene of interest, or you can say the desirable gene or foreign gene or alien DNA, or you can say the gene what we require to make the changes into our host cell. Are you getting that? This is a full DNA, but we don't want the full, we need the fragment putting into the vector, through vector into the host cell, right. Now come to the point that what do you mean by this restriction enzyme? If I take the term restriction enzyme, from where we are getting such type of enzymes which you call it as a molecular scissors, right. What you call it as molecular scissors, right. Now this we are collecting from the bacteria, right, and first discovered was Hind 2. That was the name of the first discovered restriction enzyme. Now we are having more than 900 restriction enzyme which are forming. We have extracted from more than 230 strains of the bacteria. Fine. So source of that restriction enzyme is the bacteria. Keep in your mind, I am talking about that tool which will help to cut the DNA of the source cell, and same scissor, or you can say that enzyme or a tool which will cut the DNA of the vector as well. I am talking about that tool. Fine. So here now we are having more than 900 restriction enzyme from how many strains of bacteria? 230 strains of bacteria. Now which scientist has studied more on this, and especially on one more you can say restriction enzyme, EcoR1. This is the most efficiently used restriction enzyme, and the scientist who use that efficiently and make first this recombinant DNA. This recombinant DNA first formed by the effort of which scientist? That is Herbert Boyer. What is the name of this great scientist? That is the Herbert Boyer. He along with the Stanley Cohen make this process of genetic engineering first time possible, right. Then after this I said you that most efficiently used is EcoR1, but due to some restrictions Hind 2 was not used efficiently, although it was discovered first, right. So it was discovered not by this Boyer, by the other scientist that was discovered, right. So that scientists were 3 scientists, Arbor, Smith and Nathan, that you need not to learn, being it is not related to your NCERT. Simply you have to learn about the Herbert Boyer. And what is the main role of Herbert Boyer here? The main role of Herbert Boyer is to study and make the recombinant DNA first time, but with the collaboration of which scientist? Stanley Cohen. Stanley Cohen knows how to take out the vector from the bacteria, especially plasmid, and how to introduce into other cell. And what this Boyer know about? Boyer know that how to cut the DNA. That thing they collaborate and come out with the first experiment to make the recombinant DNA. So Herbert Boyer is having big role and having important questions in your NEET exam regarding his work. So coming back to this point, if I am taking EcoR1 now, Let's talk more detail about its nomenclature. If I am talking the nomenclature of EcoR1, how we are naming this restriction enzyme. Number one, can you see the first word is E. This is derived from the genus name of its source. What is the source of that? That is Escherichia coli, right? So first letter is coming from its genus name, next two letters is coming from its species name or specific epithet, right? So coli is its species name, where CO, the two letters are given to the enzyme name. And what this R stand for? R stand for the strain of that. It is RY13. That is a strain, means variety of that bacteria. That is RY13. This is 13 here. And what 1 stands for in Roman? It is the series of extraction. Means we have to name the restriction enzyme in this way. Fine. So this is about its nomenclature. Any of the restriction enzyme. How many restriction enzyme we know present? More than 900. And we have to give them different names based on the source, based on their strain and the series of extraction. We are giving them the nomenclature. Now let's see why you call it as restriction enzyme. What is the reason behind that? If I am saying the word restriction, means it is restricting something, to understand its name, you should understand we are using the enzyme from the bacteria as a tool in the biotechnology for welfare of human. But you have to find out what this enzyme is doing in bacteria. Due to that reason, it is getting its name restriction enzyme. I repeat this line, this is very important, that why it is getting the name restriction enzyme. You are using this enzyme for our own benefit in biotechnology, but from where you are taking? You are taking it from bacteria. But to get to its name, you should know why bacteria is secreting that. It is not for your welfare. It is secreting for its own welfare. How? Like suppose this is a bacteria. It is sometime affected by viruses. Right? That viruses, you call it as bacteriophage. And that bacteriophage is having its own DNA. And that viruses, you know that they cannot live independently. They will be non-living. They are like a dust particle in the air. When they are attacking on any body, like our body or plant body or any microbes, then they will start using their machinery and start multiplying. Then they become living. Same way here, the attacking organism here is a bacteria. Right? And who is attacking on that? Virus. That virus name is what? Bacteriophage. And you know that this bacteria is having its own DNA as well. It is having its own DNA. But after some time what happened? This virus will give inside its DNA to control on its machinery of the bacteria. When it is going inside the body of bacteria, bacteria want to resist that thing. They want to stop the multiplication of virus in the cell of the bacteria. Now how it will do that? It will do that with the help of the two enzymes. What are that two enzymes? What is the work of that two enzymes? From there, first enzyme is going to protect. One enzyme is going to protect its own DNA. One enzyme, I repeat, it is going to protect its own DNA, means DNA of the bacteria. Why they need to cover its own DNA? Because once this DNA is entering inside, of virus, once it is entering inside, then what it is doing, I already said you, it is using its machinery. Then what the second enzyme will do? The second enzyme of bacteria will start cutting this DNA. DNA of what thing? Of virus. If virus DNA is going to be cut, obviously it will be no more functional. It cannot multiply in bacteria. Means bacteria have been survived by using these two enzymes. Number one, they have to protect their own DNA. Why they have to protect its own DNA? Because second enzyme which is about to release is for cutting the DNA. If they will cut its own DNA, then how they will be protected by the DNA of viruses? So can I say they are restricting the growth of viruses inside their body. So naturally they are secreting the enzyme, the second type, which you call it as a restriction enzyme. That's why this enzyme got its name restriction enzyme, because they are restricting the viruses to get multiply in the cell of the bacteria. I hope so you get this point, why restriction enzyme is getting its name. And what is the source? Bacteria. The restriction enzyme name is not based on the use what we are doing in genetic engineering. It is based on how bacteria is protecting itself from the viruses, right? So this is about a small history after its name. Now coming to the next point, if I discuss with you the types of the restriction enzyme. Basically the restriction enzymes are of two types, and first of all it is coming under the category of the nucleases. A major group of enzyme, the heading what we are giving, nucleases. What are its two types? Exonuclease and endonuclease. So what restriction enzyme belong to which class of enzyme? Nucleases, which is coming under hydrolases actually. So two types here are exonuclease and endonuclease. Restriction enzyme, suppose this is a DNA, this pen in my hand, I am considering this is a full DNA. If restriction enzyme will cut in our biotechnology, right? In biotechnology, when we are using this restriction enzyme on the source cell DNA, if it is cutting it only from the edges of the DNA, that you call it as exonucleases. When they are cutting from the within, I must say within the DNA, then you call it as endonucleases. Then most common is what thing? Most common is the endonucleases. That's why sometimes restriction enzyme we call it as a endonucleases, restriction endonucleases. So let's see from here, exonuclease is what? Removes nucleotides from the end of the DNA. And endonuclease is what? Makes cut at the specific position within the DNA. Right? So two types are there of that, exonuclease and endonuclease. Fine. Now after this, let's talk about its functioning, how it is function, how it is reading the DNA and where it is going to cut it. This restriction enzyme for its working, will read the full DNA. Of which cell I am talking about? Source cell. From there you have to remove the DNA. Now if you are keeping that in restriction enzyme, it will read the full DNA, and where they get a appropriate site or a sequence of nucleotides, at that particular point it is going to cut it. For example, if I am taking for EcoR1, it is cutting at which specific part? That you call it as a recognition sequence, right? And that recognition sequence usually made up of 4 to 8 base pairs. And out of 4 to 8 base pair, common is what? 6 base pairs. And for EcoR1, which is an enzyme, will be cutting at which recognition site? GAATTC. where this sequence will come, or this is I am taking 3 prime to 5 prime, opposite to that what? GAA TTC. This is like this. If I am taking this sequence in this way, like let me to show you from this point, like even in your book you will find this picture showing you in the DNA we are having some sequence specific GAATTC. This sequence will repeat after some certain set of the base pairs. When it is repeated, echo R1 will cut only at that specific point. Let me to recall that thing, we don't have one restriction enzyme. We are having many restriction enzyme. They will have different recognition sequences. And that chart, let me to show you once here. This is like this. These are the name of the enzymes. And this is the name of the source. Like for example, I said you echo R1. So the source name is what? Escherichia coli and strain RY13. Fine. And this is its recognition sequence. Likewise, other restriction enzyme is having their specific recognition sequences. And they are cutting at specific point. And usually where it is cutting, let me to take this point here. Usually they are cutting at the point away from the center. Can I say center of this sequence is like this? Let me to complete this part, or let me to take on the another slide. What is the sequence? GAATTC. GAATTC. Why I am writing twice? Because you know that DNA is double stranded. We have to see that sequence on both the strands. Fine. But how to read that sequence, I will take it later on. But let's see how it is cutting such sequence. In that, can you see how many base pairs are there? 1, 2, 3, 4, 5 and 6 base pair. At this recognition sequence, they are going to cut the DNA. I said you the point of cutting mostly will not be from the center. It will be away from the center, slightly away from the center. Like for this, the cutting will be from here, from here. Can I say between purine and purine for this echo R1, right? Between purine and purine. So it will be cutting it like this. It will be cutting it like this, slightly away from the center. And after partition of this, what you will get? You will get GCTTAG, and opposite to that you will get AATTCG. Can you see, like a purse is open like a clutch? It is going to open like this, right? It will be open like this, and you will get unpaired. You will get the unpaired nucleotides. That unpaired nucleotides, what you call it as? You call it as sticky ends, or you call it as staggered ends, or even you call it as cohesive ends, or even you call it as overhanging stretches, right? These names have been given if the restriction enzyme is not cutting from the center, away from the center. From the segregated part, you will get unpaired nucleotides, and that you can say paired nucleotide, that end you call it as overhanging stretches, sticky ends, staggered ends, or cohesive end. Fine. Now let's see the sequence once again. Can you see the sequences GAATTC? I repeat here, GAATTC. If we will carry this process according to your NCERT book, there you will find this sequence they have given in some specific sequence. Like if I am assuming 5 prime to 3 prime, this even 5 prime to 3 prime. It is the thing when you are reading one strand, the direction, see the polarity I am taking 5 prime to 3 prime. What it is coming? GAATTC. Again from the opposite strand, the polarity must be same, 5 prime to 3 prime. And same the sequence will be GAATTC. That type of recognition sequence or that recognition sequence you call it as palindromic sequence, right? Such sequence you call it as what? Palindromic sequence. So this recognition sequence are palindromic. If you see some English word like or a language name Malayalam, when you read like this, the letters will be MALAYALAM. If you are reading like this, again it is MALAYALAM. There is no change in the reading of the letters, but this is happening in same word, but this is not happening in DNA. You have to read both the strand. Although the sense is coming same in front go or in from back to front, but the polarity on the two DNA strand must be same. If you are reading this from 5 prime to 3 prime, opposite strand must be from 5 prime to 3 prime, then you will get the same sequence of the nucleotides. Fine. So this is about the recognition sequence where they are going to cut, and that is a palindromic sequence. Now let's see on the host DNA how they are doing the cutting. Suppose this DNA is a foreign DNA you have taken from the source cell, and this is what thing? Vector. In the beginning, if you remember, I said you that we have to take like here I said you we have to take the source cell, and from source cell DNA you have to remove it out and take this piece and put it into the vector. So in that diagram just now what I am going to show you, it is showing the part of DNA carrying this desirable gene, and one part is showing the part of the vector DNA which you have to cut there, then further this gene is going to be inserted. Fine. So let's see that flow chart, that's very important to understand that concept. So this is what thing? Vector DNA, right? And that after cutting you will get, let me to show you this point better first from this diagram. I hope so you will get a better view when you will see this thing, then I will go in a microwave. This is the foreign DNA which you are taking from the source cell, and this is which DNA? Vector DNA which you have taken from the bacteria. You are using the same scissors to cut it. Here what they have used? EcoR1. And what is the recognition sequence for that? GAATTC. Both it should be here and even there, right? Now if you are gene of interest is here somewhere, and you will find in the gene of interest surrounding that, this is suppose a gene of interest, surrounding that the base pair is having recognition site GAATTC. I repeat, if your gene of interest is surrounded by both the sides with the recognition sequence GAATTC, then which restriction enzyme you will use? EcoR1. Then that same you have to use for the vector. Fine. So here the sequence we found GAATTC, so we are using EcoR1. It will be cutting from here, even it is cutting from this point, right? Now what it is cutting, which bond break, in the next slide I will explain. Just now the cutting happened and you will get this gene of interest, not much easily, much more steps we have to perform, but you assume we got this gene of interest. And parallelly you are cutting what thing? Plasmid with the same, and then this must be pasted in the vector DNA, and you will get this recombinant DNA, right, which is carrying the gene of interest, right, or which you call it as alien DNA, or you call it as foreign DNA, Now we are using the fragment of that, simply we can call it as DNA fragment. And what is this DNA? This is the DNA from the plasmid of bacteria, or you can say the vector. Fine. And then you are keeping that into the cell. Which cell? Host cell. This host cell back can be the any other bacteria which is not carrying the desirable gene of interest, right? It can be plant cell, it can be animal cell. And that process you call it as what? Transformation, because it will change the phenotype of the host cell. What earlier it is not showing, now it will be start showing due to the presence of this foreign gene. And when this host cell will divide, that process you call it as that multiplication or retaining from generation to generation to generation, that gene of interest. You have to maintain it, maintaining that gene of interest from generation to generation when the host cell is going to be divided, right? So this is the thing, at this point we have to use it and bring it out from the foreign DNA and putting into the vector, making recombinant transformation and maintaining that thing from generation to generation. Now let's see on the micro level. If I will put a view expand on this DNA of the foreign and here on the plasmid, then what sequence and staggered ends we will get there, like this, which I opened earlier. Now it is useful for you to look into this picture. Can you see this is a vector? Now I have shown you the vector on that side. In this picture it is vector DNA from where you have cut it GATTC from this point away from the center. Keep in your mind this is a vector DNA. Here we have taken foreign DNA, right? Only one end they have shown for vector, one end they have shown for the foreign DNA. How? Like see back again if you cannot able to understand. Like here is the which DNA? This is a gene of interest, and you are taking this point where this gene of interest is going to bind with the vector DNA. You are showing this part. When this is a vector and this is a foreign, they are going to bind, right? It is binding on both the side like this, right? I repeat, this is a foreign DNA and this is a vector. So they are binding on this point and even on this point. So we are showing you only one point out of it, right? So what is that binding point which is having GATTC and sequence reverse, same sequence for foreign DNA as well as for vector. And if you are cutting it like this and here you are cutting it like this, can you see you are getting unpaired nucleotides? What I said you already, these end you call it as which end? Sticky ends. You will call it as sticky ends, both here as well as here. When you are cutting with same restriction enzyme, obviously same, or you can say the same recognition sites are going to be cut and you get complementary nucleotides opposite to that. When you try to paste it, and that pasting is done by which enzyme? Ligases. Here I am introducing one more tool for the genetic engineering, which you call it as molecular glues. This you call it as molecular glue. Like glue is there to paste something together. So ligase is putting together what thing? This one, the vector DNA and the foreign DNA. And who is cutting that in the beginning? Restriction enzyme, which you call it as molecular scissors, right? Now let's see what they are cutting actually. This is actually what thing? Can I say this is all about the nucleotide sequence which is binded by which bond? That is binded by phosphoester bond. By which bond they are binded? Phosphoester bond. If they are cutting from this point, that means which bond is going to break? Phosphoester bond. Same here. What bond is going to break? Phosphoester bond. And it will break the hydrogen bonds as well in this region, both at this point as well as on this point. Then you get this complementary sticky ends of foreign DNA and vector, and you bind it and you make a recombinant DNA. Let me to explain you through one big layout, GAATTC. GAATTC. Where it is cutting? On this point. If you are cutting like this, right, what is this point of cutting? Can I say this point is breaking which bond? This is cutting the phosphoester bond. And this line is indicating which bonds are going to break. This line is indicating the hydrogen bonds are going to break. Check it out, how many hydrogen bonds are going to break here? 1, 2, 3, 4, 5, 6, 7, 8. Because adenine and thymine are having 2 hydrogen bonds. So how many are going to break? That 8 hydrogen bonds are going to break. And how many phosphoester bond? 2 phosphoester bond is going to break at 1 recognition site. Right? So this is the way they are going to cut the recognition site. I repeat, the recognition sites, they are having some base pair. Think your desirable gene of interest is somewhere here, but that gene is surrounded by some specific recognition sequences. You have to check what recognition sequence is there. If suppose the recognition sequence is this, then you need hind 3. If the recognition sequence is this, then you need the PST1. Now we have discussed the full story based on this recognition site whose enzyme is what? EcoR1. Then you have to use that thing, and that will not cut only on that point, it will cut the full DNA, it will read the full DNA where this recognition sequences are there, they will cut it and you will get the small fragments or big fragments of the DNA. And you will make it separate by next technique, gel electrophoresis. I now talking only about the main tool here, that is a restriction enzyme, which is more important, or you can say according to your NEET exam, right? So based on the recognition sequences, we are having the enzymes. But what enzyme you are using for the source DNA? You have to use the same enzyme for the vector DNA as well. This is more important, only then you will get the sticky ends. And it is not like that always we will get this sticky ends and we will get unpaired nucleotides. When you will get unpaired nucleotides? When it is cutting away from the center. But some enzymes are there which are cutting it from the center. Can you see here SMA1? When you will see its, you can say sequence, CCCGGG. Opposite again, being it is palindromic, for all the recognition sequences are palindromic. So it will be sounding same here as well. Then where it is cutting actually? It is cutting now from the center. It is going to cut from the center, from here. If it is cutting from the center, can I say this part will be going together and this part is going separate? All separated part is going to be bonded. There is no unbonded base pair. There is no unpaired nucleotide. Such type of ends if we are getting, such type of ends you call it as a blunt ends, or you call it as a even ends, or even you call it as flush ends. These are the names if the restriction enzyme is cutting at center, which is very rare, very less, but mostly they are cutting away from the center of palindromic sequence, right? So this is the way how our tool will work in the biotechnology and help us to get the better of the organism, to get the best product from there, right? And they are going to cut the DNA for the foreign as well as for the vectors, same we are using. But how many varieties we are having, means how many types we are having, that is more than 900. From how many strains? More than 230 strains of the bacteria. And which is the first discovered restriction enzyme? HIND2. and which is more efficiently used and worked by the Herbert Boyer, that is Eco R1. Fine. So this is the most important tool we have taken for our today's discussion. I hope so you have understood that. Please do revise it, and after revision do try your multiple choice question. This is all for today. Thank you, students.